Great. Hi, everyone. Can you see my screen now? Great. It's nice to meet everyone. And my name is Gan Li. I'm assistant professor in the department. It's a pleasure today to talk about some recent work on microbiome data analysis. So this is a joint work with PhD student Yan Li and my collaborator Quinn Chen from UConn. So microbiome is really everywhere, and human microbiome is in and on every inch of human body, and it's a big component of us. Studies have conjectured that microbial cells and genes may outnumber the human cells and genes. So to some extent, it's fair to say that we are more of the microbiome on and in us rather than ourselves. And microbiome is not static, it's dynamic, and it's changed all the time and under different conditions. So that's why microbiome has been shown to be significantly associated with many complex diseases, such as diabetes, cancers, and even neurological disorders. So people have used microbiome profile to try to do prognosis, diagnosis, and even come up with new therapeutic interventions. So it has been a very active field and a burgeoning field with a lot of challenges. So this is a diagram adapted from a paper in 2013, just showcasing how microbiome data are typically acquired. Basically, samples are extracted from human bodies and go through this amplifier, and then different type of sequencing procedures are conducted on these amplified samples. In practice, there are two mostly commonly used sequencing procedures. One is the applicon sequencing, also known as the 16 rRNA sequencing, and the other is a shotgun sequencing. Afterwards, a sequence of bioinformatics steps are involved to get what we call the OTU data table, the operational taxonomic units. So this is a data table with samples and features. So features are named as OTUs or taxa or microbes. I'll use these terms interchangeably in this talk. So basically, it's a data matrix of samples and features. Once this table is obtained, various analyses can be conducted on this table, such as quantification of diversity, data visualization, differential abundance analysis, network analysis, etc. And today I'm primarily focused on regression analysis. And our goal is to associate this OTU data table with some clinical outcome. And I'm going to show the challenges and opportunities with this type of analysis. So you may wonder why we can't just use some standard linear regression or more complex regression models that we have in the literature to study this problem. Well, there are many challenges associated with microbiome data analysis, and here are some unique challenges. For one thing, we know that microbiome data, different samples usually have different library sizes. In other words, the total read counts. So a very common way to normalize data in microbiome studies is to convert all the data to compositions. In other words, we divide the read counts of each taxon or each microbe by the total number of read counts in that particular sample and make it a compositional vector. So for each sample, the compositions of different taxa sum to 1. So this compositionality is a common feature in microbiome studies. So this is one unique feature. And once the data is normalized as compositions, then it's no longer admit the standard Euclidean geometry. Many standard notions that we know in statistics does not work anymore. So this is one challenge that has to be conquered. Another is that there usually exists taxonomic or phylogenetic information between different microbes among the microbes. So the taxonomy and phylogeny can be represented as tree. So shown here, you can see this is a phylogenetic tree structure among the microbes. So this information is commonly known as extrinsic information for the data. And also the data is typically inflated with zeros. Some zeros are due to the structural, some zeros are structural zeros, meaning really certain microbes are absent in the sample, while some other zeros are basically due to the sampling area. And also the data measured with a lot of error, and the dimensionality of the microbes usually far exceeds the sample size. So all these present unique challenges to this to the microbiome analysis. So we cannot simply apply the standard linear regression to this microbiome data and get what we want. So we have to come up with some new ideas. So in the literature, the most commonly used regression model for microbiome regression is the log contrast models. So these models are developed in the 1980s by Hutchinson and co-authors, where the basic idea is very simple. Essentially, they pick one of the taxa as the reference and then use the log ratio transformation to convert the original compositional data to remove it from a simplex to the Euclidean space. So the general form of the log contrast model can be viewed like this, where the response is regressed on all these log ratio transformations. And easily you can see that there is an equivalent symmetric form of this model. Basically, you can rewrite the model as a regression model on this log terms subject to a linear constraint, that the sum of the regression coefficient is 0. So these models are very commonly used in the literature, mainly because of the fact that this log transformation can remove the compositions from the simplex to the Euclidean space. So it shares many nice properties that we can still enjoy in this Euclidean space. And some other transformations are also used in the literature similar to this, but slightly different, such as the center log ratio transformation. So this is the main school of thought in the microbiome regression. However, there are certain limitations of these transformation-based models. For one thing, you can easily say that these log transformations are inadequate in accommodating excessive zeros. So we cannot apply log transformation to 0 values. So the common practice is to replace the zeros with some arbitrarily small value, for example, half of the detection limit, to replace the zeros in order to avoid singularity. But apparently that will introduce bias, and sometimes that bias can be very severe because we have excessive zeros in the data. And another limitation of this type of methods is that it lacks straightforward biological interpretation. So as you can imagine, even if we apply this log transformation, the compositional data, they are still interrelated. In other words, we cannot simply interpret the regression coefficient as the effect on the response by changing the corresponding predictor by 1 unit. Because if you change the one predictor, you know for sure that some other predictor values will also be changed, because before the log transformation, they sum up to 1. Right? So it lacks straightforward biological interpretation. And also it hinders the incorporation of extrinsic information such as the taxonomy or phylogenetic information. And finally, if we apply this model at different taxonomic ranks of the same data, we may get very different results. There's no guarantee that this model gives you compatible results across different taxonomic ranks. So these are all the very realistic limitations of these transformation-based log contrast models. Then our question is, can we come up with some alternative way to model this compositional data? More specifically, can we directly model them without this transformation? And the answer is yes. And that's what I'm going to talk to you today of our new work. So we propose a new paradigm for compositional regression, what we call the relative shift model. So we start from a very simple setup. Consider the very simple linear regression of y regress on this x1 to xp, so p-dimensional compositional vector. So this is perhaps the simplest model that we can ever come up, where x1 to xp, they are compositional. Okay? They sum up to 1. But right away, you can see that this model is not identifiable because they are compositional. So we noticed that after reducing the degree of freedom by 1, actually we can achieve identifiability right away. In other words, if we just eliminate the intercept term, this intercept-free model linear regression actually is identifiable in the sense that there's no identifiability issue. And moreover, we find that actually this simple model will have many nice properties in practice. For example, it has superior interpretability in the way that the contrast of any regression coefficient pairs or triplets or quadruples can be interpreted as the relative effects of compositional shift. For example, take this pair of composition as an example, J and K. Okay, you can simply rewrite this sum as beta K times the sum of these two compositions plus the contrast of these coefficients multiply xg. In other words, the contrast of beta J and beta K can be directly interpreted as the effect on response Y by shifting a unit concentration from the K's taxa to J's taxa while holding all the other taxa concentration fixed. Okay. Similarly, you can show that any contrast can be interpreted in this way. So in other words, the contrast of regression coefficients can be interpreted as the relative effects on concentration shift. That's why we call this the relative shift model. And right away, you can also say that it enjoys some combinatorial property. In particular, if the regression coefficient for the J's taxa and K's taxa are the same, then these two taxa can be combined without losing any information. These also serve as the basis for incorporating a taxonomic tree structure later on. So, and also this model has scale and shift invariance property, meaning if we add or multiply a constant to this response variable, we can simply do the same for all the regression coefficient and the model stays the same. Okay, so in other words, the absolute value of this regression coefficient does not matter, does not have a particular meaning, but the relative relation between different regression coefficients actually matters. So this is the basic relative shift model. And directly comparing this model with the log contrast model, you can see that they are very different and built on very different rationales. In particular, in terms of predictors, we do not use any transformation, and the coefficients do not need to, we do not need to impose any constraint on the coefficients. And in your interpretation, we can directly interpret the contrast of regression coefficients as a relative effect. And in a second, I'll show you that it's actually very easy to incorporate extrinsic structures such as the taxonomic tree. Okay, and it's also robust against measurement error, as I'll show you in a couple of slides. So, talking about the taxonomic tree, this hierarchical structure actually is very commonly, very easily obtained for microbiome data. It captures the tree structure relation between different variables or features in the microbiome data. For example, these figures show you the taxonomic tree of 62 taxa in our real data example. As you can see, most taxa are at the genus level, while some other taxa are obtained at the family, order, class, or phylum level. Okay, so this is what the data, real data typically look like. And also you can see that different number of genera may combine to form a new family, and different number of families may form an order, and so on and so forth. So this information is commonly available, readily available in practice. And in general, people have seen that it's useful to incorporate such information in the analysis, various analysis. And here, our goal is trying to incorporate such information in the regression analysis and say how we can leverage this information. So consider our model. Recall that the basic idea is that taxa sharing similar taxonomic path are more likely to be combined. In other words, if two genera share this belongs to the same family, then they are more likely to be combined without losing information than two genera that belongs to different families or different orders. Okay, so that's the basic idea. And recall that our relative shift model, combining taxa is essentially equivalent to equating regression coefficients. So all we need to do is to come up with a way to translate the original taxonomic tree to some equisparsity regularization. Okay, once we have that equisparsity regularization induced by the tree, we essentially can impose or incorporate that tree structure into the regression. And to achieve that, we come up with a new tree structure regularization. Essentially, the idea is as follows. The first step is really just to reparameterize the original parameters, regression coefficients beta. So we borrow idea from a recent publication that is to break down this regression coefficients beta, the original regression coefficients, to intermediate regression coefficients gammas. We assign each node an intermediate regression coefficient along the tree and require that the sum of the intermediate coefficients along the way to the node, to the root node, to be equal to the original regression coefficient. For example, beta 1 here, which is the regression coefficient for the original, for the first taxon, is equal to gamma 1 plus gamma 8 plus gamma 10 plus gamma 12. Okay, so that's how we reparameterize the regression coefficients along the tree. And the next step is to introduce the overlapping group Lasso penalty on this intermediate coefficients, where each group is induced by the descendant nodes of each internal nodes. So if you look at the right panel of this illustration, as you can see, so essentially for each internal nodes, we form a group for all its descendant nodes and put a group penalty on this descendant nodes. As a result, if one group is equal to 0, then the corresponding coefficients, corresponding to those leaf nodes actually are equal to each other, so that we achieve equisparsity. Okay, so take this as an example where gamma 9 equal to gamma 5 equal to gamma 6 equal to gamma 7 is 0. Then by definition, we can say that beta 5 and beta 6 and beta 7, the three regression coefficients are equal. Okay? So that's how we introduce equisparsity. So putting everything together, we come up with this regularized least squares, where essentially the square term is a loss function on the original coefficients, and the penalty term is on the intermediate coefficients. And these two parts are connected by this linear constraint, where beta is equal to A times gamma, where A is a known matrix induced by the tree. Okay, and this we can easily adjust for covariate effect by incorporating covariate as additional covariant matrix. And this optimization is a convex optimization and can be easily addressed by a smoothing proximal gradient algorithm that I won't go into detail. So the tuning parameter can be selected by cross-validation. So there are several key features with this taxonomy regularized relative shift model. One is that it can achieve equisparse coefficient estimation, which directly translates to feature aggregation. Okay? And in particular, since we aggregate features based on the taxonomic tree, at the end of the day, we will be able to select different entities at different taxonomic ranks adaptively. In other words, a byproduct of this estimation is to achieve taxonomic rank selection. In other words, we can identify the taxa that are important at different taxonomic ranks. And also, since we use information across the taxonomic ranks, these results are compatible across different ranks, and it achieves good balance between data resolution and accuracy. We also provide some finite sample prediction error bounds for the estimator. And under certain scenarios, we can show that this actually is further bounded by the number of unique episparsity families. So this is a very classic balance that we can achieve that connects to many standard regularized regression methods. Okay. So for the interest of time, I'll omit the simulation study where we show that this method actually outperforms many competitors in many relevant settings. I'll just directly jump to a real application to a preterm infant neurodevelopment study and showcase how what is the performance of this new method. So this is a study of a NICU neonatal intensive care unit preterm infants neurodevelopment study, where in the literature, people have shown that gut microbiota may be associated with cognitive behaviors through the so-called gut-brain axis. So this study is really trying to corroborate those findings by collecting samples from 34 preterm infants during their first month of postnatal age. So we use 16S rRNA sequencing procedure to obtain the data, the microbiome data on 62 taxa up to the genus level. So recall the taxonomic tree that I showed you earlier. So that is the taxonomic tree among the 62 taxa. Our response is the neurobehavioral outcomes, which are measured by the NICU Network Neurobehavioral Scales score, the NNS score. So our goal here is try to associate this NNS score with the microbiome data after adjusting for additional covariates such as gender, birth, weight, delivery type, and complications. So we apply our method to this data set. Although it's a small data set with only 34 samples, we still get very interesting result out of it. So this is the parameter estimation result after cross-validation. As you can see, the taxa with the same title colors are clustered or are aggregated. So for example, this taxa 2, taxa 3, taxa 4, they share the same regression coefficients, so they're aggregated up to the outer level. Okay, as you can see, this is a demonstration of the parameter of the regression coefficient that we can get from applying our model. So you can say that different taxa are selected at different taxonomic ranks adaptively. Some are aggregated up to the class level, some are aggregated to the outer level, and some remains at the genus level. Okay, so we compare this result with some previous analysis that we conducted on the same data set, but strictly focusing on the outer level, and we see consistent result. In particular, I want to highlight two orders that are very interesting to see. One is this, which is the Clostridios. It's taxa 24 to 38, and previously we identified this to be a significant predictor of the NNS score. Now we also select this to be significant, but what this new analysis have shown us is that this order is important as a whole, while the individual composition of its children or descendants does not really matter. You can arbitrarily shift concentrations between different genera or families, but as long as you maintain the concentration of the order, then it has the same effect on the response. Another order is this one, the Enterobacterials. So from this analysis, we say that actually for this one, individual genera in this order actually matters in terms of predicting this NNS score. So this result provides consistent result as our previous analysis, but provides much richer information. Furthermore, we conduct some quantitative comparison with existing methods using leave-one-out cross-validation. In particular, we compare this leave-one-out prediction mean square error with some state-of-the-art methods, such as the log contrast model with lasso penalty, the kernel penalized regression with different penalties. So this is the result. These are the prediction accuracy based on the leave-one-out cross-validation. So although they're not significantly different from one another, mainly because we only have 34 samples, very small sample size, but there are some suggestive trends from this result. For one thing, we say that the proposed method, although we use a very different paradigm, regression paradigm, compared to the existing state-of-the-art transformation-based methods, they have similar result. So the new paradigm is at least comparable to the existing ones. And another finding is that using this taxonomic tree structure actually improve the prediction performance. So if you say these results, the best two actually is our method and the KPR tree, which both use the taxonomic tree structure. So the other two does not use, so they have slightly worse performance. So these two messages are quite suggestive. So for the interest of time, I'll briefly summarize. We come up with the relative shift model that directly use compositions as predictors, and the coefficients capture the relative effects of shifts in concentrations. So this new regression paradigm provides a flexible basis to incorporate additional structures such as a taxonomic tree, and this new tree guided, and we propose a new tree guided regularization for feature aggregation. And as I mentioned, this is a whole new regression paradigm for compositional data. There are a lot of additional stuff that needs to be done. For example, how to incorporate the phylogenetic information, how to model the nonlinear relation between the microbiomes and the response variables. So those are all open questions. And I'm happy to bring on board motivated PhD students or graduate students to work with me on this. Okay. All right, so I'll just stop here. Thank you.
Thanks, Ken. So I guess we'll take, if there's any questions, if you feel more comfortable to ask your question through chat, I can read it off for you, or you can just unmute yourself and ask. We'll take maybe just a couple just for the sake of time. I don't want to go too past 4.
Hi, Ken, this is Pei Song. I have just a couple of very quick questions, I think.
Sure.
So one is, so you establish some theory, the error bound, right? The finite sample error bound, I guess. So then in the error bound, does not the sample size, but the number of covariates play a role in your error bound?
Yeah, that's a very good question. So I didn't go into detail here, but the number of covariates does not matter, but the number of unique equisparse groups matters.
Okay, I see.
So that's similar to the standard sparsity regression problem where the number of non-zero matters rather than the total number of predictors.
Okay. So another related very quick question is, so in your data application, what is the number of covariates or number of groups?
So we have 62 taxa in this example, and 7 to 8 covariates on 34 samples.
Okay. In your application, do you have any general guidelines regarding the number of covariates or number of?
It doesn't really matter as far as we are concerned. So as you can see, this is this can be considered as a high dimensional problem because we have more features than samples, right? So with this equisparse penalty, essentially you can handle much more variables than samples. So there's really no constraint in that perspective.
Okay, thank you.
Yeah.
I saw 2 other questions in the chat. I think Kathy's question was the same as Jonka or as Pei Song's, and which was what's the usual P that you encounter? And then Jonka asked, have you thought about incorporating tree estimation uncertainty based on the fact that I guess you're taking the tree as given right now, but
right.
Of course you have to estimate that tree. And so how or has anyone thought about trying to incorporate that uncertainty into your estimation?
That's a great question. So short answer is we haven't, mostly because in microbiome studies, basically, at least for the taxonomic tree, there's really not so much uncertainty. So it's not really estimated from data. It's given by some prior biological information. So that is something that we can safely consider as given. But for moving forward, if we want to incorporate a phylogenetic tree, I think there is more uncertainty there. So definitely incorporate some uncertainty from the tree structure will be of interest to explore.
Hi, Gen. This is Jian. I have you have a very nice talk. Thank you. So I have a very basic question regarding to interpretation of your model. So can you interpret a single beta J, or you have to specify the reference category to interpret beta J?
Yeah, that's a very good question. So we cannot interpret each individual beta J. As I mentioned, each individual beta J, the value of that does not really make much sense because of this scale and shape invariance. You can arbitrarily add constant to the response or multiply by a certain constant, add or multiply by a certain constant, and then correspondingly, all those coefficients will shift by the same constant. So the absolute value does not make sense. What we care about is the relative relation between different coefficients.
So then when you do the estimation, you got the sort of estimate for each beta. So you always need to interpret them on the relative shift. So
yes,
if you do statistics,
right, you can only interpret the contrast of the regression coefficients. So that's why we don't incorporate any like Lasso penalty or whatever. So we only encourage equisparsity. So we don't care about the individual value of the coefficients.
I see. So for your theoretical work, what is the condition on this X design matrix? Do you have any condition on the sort of sparsity on those X?
No, for X, we just require to be compositional. That's all.
So, but if you have like two column all equal to 0, you cannot handle that, right? At least there has to be non-zero. At least has one element has to be non-zero.
They have to sum up to 1. So for each sample, they have to sum up to 1. So you can have 0.
But if you have a lot of zeros in your category, still you're still able to show these results. But when you do the model fitting, maybe there are some potential problems.
Yes. So if you have, for example, if for several taxa it's all 0 for all the samples, essentially you should drop them, right? You should not incorporate them in the model because it doesn't really make sense to incorporate them. It doesn't provide any information, right?
Right.
Yeah.
Okay. Thank you.
Thanks.
So there are 2 other questions. I guess we'll say that we'll do them very, very quickly. So again, if you can give maybe like a 30-second answer to each. So G asked, it looked like you had different overlapping groups of different sizes. Did you consider that in your penalty? And then Jeremy asked about interpreting beta equal to 0, which I believe is related to Jian's question about the relative shift.
Okay, so first to Jeremy's question, beta equal to 0 does not mean anything in this model. We only care about the relative relation between different regression coefficients. So that's my short answer. And to the first question, that's a very good question. So we can incorporate additional weights in front of each group to balance different group size, but we choose, well, it's still in progress. So we haven't decided whether that's a good practice or not. So at least from the theoretical point of view, we don't need that. But that's a good question. Thank you.
Thanks, Ken. So if you have more questions for Gen, I guess we can save them for later, and we'll just move on to Jean's portion of the seminar. Jean, why don't you take it away, and I'll just let you go with it.